Right. So then I will move on to the next question that I tend to find people ask, and that is that are there any problems with the injection method? So we're talking about the injection method here with regards to the pressure that one is applying. So in other words, do you rupture the heart commonly when you do this? Now, so Matt has actually done some great work, and this is in the paper, and these figures you can find in the paper, where he's actually looked at the ability to isolate cells in terms of the percentage of rod-shaped cells, but also in terms of the total number of rod-shaped cells that you get with using different flow rates. So, and obviously that to some extent relates to the pressure as well. So in other words, and what he's really found is that increased flow rates, i.e. pressure, presumably, do not really substantially affect the yields of either the percentage of the rod-shaped cells or the total numbers of rod-shaped cells. And Matt might want to comment more on this. But equally, some of the more recent work, which is not published yet, is been looked at the MI models. So the artery ligation was used to induce a myocardial infarction, and in 8-week-old mice, and you can clearly see that this region here is not been perfused very well, so it's not stained with a dye, and then the cells were isolated from those hearts which have an MI as well, so therefore a thinner part of the ventricle as a result because of cell death. And as we would expect, whilst the total cell numbers in the MI hearts are reduced compared to the shams, as well as the total numbers of total percentage of rod-shaped cells is reduced, actually you were still able to successfully isolate cardiac myocytes really without rupturing the hearts at those infarcted regions. So based on the evidence and our experience so far, I would say that rupturing the heart using the injection method is not a common thing that tends to occur. Although, as we probably discussed earlier, probably you should be targeting these flow rates of 1 to probably 2 to 3 mils per minute, because that seems to be improving your success of cardiomyocyte isolation, actually using both the injection method as well as the ligation-induced method, as we've discussed earlier. So that's probably how I would answer these types of questions. That's our observations from work in our labs, in Matt and Roger's lab in Singapore as well. So I don't know if Matt and Rongli have any further comments to make based on this in response to this question.
I think, first of all, I can't take personal credit for this flow rate chart. This was done using automated systems, kind of painstakingly set up by. We have a fantastic lab tech called Peter Lee, who was done all of this work. And I just counted the rod-shaped cells afterwards. That was my role in that particular.
It's an important contribution.
Yeah. I'm trying to tell myself that. We were surprised actually that when you increase the flow rate that much, we still seem to get myocytes. So I think like we mentioned before, it just wasn't really necessary. The heart didn't rupture, but I think it's best just to, using the injection method, it's best just to inject as low a amount as is necessary to ensure that the heart is fully inflated, and that really, you don't need to go above that. As for rupture, I'm trying to think now how thin our heart walls got after the myocardial infarction that we did. They certainly weren't the thinnest that we've seen in some cases where other people do myocardial infarction and the wall becomes very, very thin.
Sure.
So I can't say that it thinned to that extent. And also, this was I think it was a week after the infarction had occurred. So by this point, fibroblasts would have come in and be depositing collagen, so that scar might be strengthened.
Sure.
So had we have isolated cells, for example, 1 day post infarction, before that strengthened scar has time to form. Again, I can't vouch for the fact that we wouldn't have a rupture problem, but we just haven't actually done that experiment yet. So
Sure.
that's all I can really add to that, I think.
But in healthy hearts, I mean, I have not really seen much rupture in our hands. I don't know if you have.
I never have, actually.
Right.
I think you'd really have to go for it if you wanted to rupture your heart, but I don't really think that's something you need to do.
Great.